All right, so now I want to talk about optimum growth. Optimum growth is when nothing is limiting growth except the organism's own biology. And so optimum growth is important in the lab just because we would like to, when we're trying to culture a bacteria, we would like it to grow as fast as we can make it grow. But also it's useful to try to achieve optimum growth to know what it is. You know, some microbes grow faster than others. I should say this. Any bacteria that's not in its optimal environment will grow slower than it could in its optimal environment. But if you could find the optimal environment for every species of bacteria, some of them will just grow slower than others because of their own biology, their own biological code. Okay? And if you want to know, so you have all these bacteria inhabiting the human body, it's useful to know who are the fast growers and who are the slow growers, not just when it comes to their optimal conditions, but just absolutely speaking. So we like to try to achieve optimum growth rates in the lab because it tells us something about the bacteria. It also makes it easier to study them if we can grow them quickly. Okay, so optimum growth is the growth rate that you see when there is nothing limiting the growth of that species except its own biology. It's got enough light, it's got enough carbon, it's got enough nitrogen, it's got enough energy, it's got the right pH, it's got the right oxygen environment. You know, if all the conditions are perfect, how fast can it grow? That's the optimum growth rate. So when you're trying to figure out the optimum growth rates, the things we usually consider in the lab are what nutrients does this microbe need? What temperature is ideal for it? What's the ideal pH? What's the ideal osmotic pressure? And that's kind of like the saltiness of the external environment, like how many solutes are there, the water pressure kind of thing, the osmotic pressure. What oxygen concentrations are perfect? So those are the environmental variables we consider the most, but even considering those, we have not been able, there must be something that we're missing, because even considering these environmental conditions, that may seem like a comprehensive list to you, but even considering all these environmental conditions, we have not been able to culture most bacteria in a lab. So DNA technology tells us there are far more bacteria in the world than we are able to study in the lab. Here is a tree of life for, here's a tree of life, and in blue you have bacteria, okay? So blue is bacteria. There are 12 bacterial phyla here in blue. These are the phyla for which we can culture at least one species of bacteria. But there are about 73 candidate phyla, which are phyla that have been shown to exist through DNA sequence data, many of whose sequences show up repeatedly and consistently in DNA sequencing efforts, but for which not a single species can be cultured in a lab. In some cases, we have so many sequences from one of these candidate species that we have been able to reconstruct its entire genome, and still we cannot observe it in the lab. What I'm saying is, most bacteria, by far, have not been successfully cultured in a lab. Culturing a bacteria in a lab is essential to understanding its biology and ecology. We can get some idea of a bacteria's lifestyle from the genome sequence because we can see what kinds of enzymes are encoded in the DNA, but you really cannot understand how a bacteria behaves without being able to observe it under different laboratory conditions. You can't see how it reacts to different antibiotics. You can't see how it reacts with other bacteria if you cannot grow it in a lab. So essentially, you can say we know very, very little still about bacteria as a whole. There are 12 phyla that we have cultured at least one species, not even all of the species, but at least one species from that phyla in the lab. There are 73 phyla that we know exist that we have not, but for which we have not been able to culture a single one in the lab. Okay, so the vast majority of the bacterial world has not successfully been grown in a lab, and we don't know why. Okay, I just want to mention that great advances have been made in culturing once unculturable bacteria by taking into account complicated elements of bacterial ecology. For example, realizing that some bacteria cannot grow in isolation, but actually require a partnership with another microbe for survival. Culturing 2 or more species together can help solve some of the mysteries of laboratory bacterial cultivation. By first culturing all the bacteria in an environmental or human sample, you can culture many that cannot be cultured alone. You can then run experiments to see which pairs of bacteria need each other. Here in this picture, so on this plate on the left, there's an environmental sample that's been plated, and all the bacteria that were in that environmental sample are all growing in a mix here. And then here, you can see it's this bacteria cultured here was once uncultured. It doesn't even have a name yet. And we have been able to culture it by pairing it with what we call a helper species. When the helper species, which is on this paper disc, is grown in the same petri dish, then you can get this bacteria to grow. But it will not grow in a petri dish alone. So why might that happen? Well, we'll get back to that when we talk about microbiomes, because when you're talking about the human microbiome, you have hundreds of species relying on each other, hundreds of bacterial species living together, relying on each other in the human body in these complex ecological webs. And so we'll talk more in detail about that at the end, but some reasons very briefly might be that this bacteria might produce a waste product that this bacteria needs to survive and can't live without. This bacteria might produce digestive enzymes that digest food in the environment. This bacteria might not be able to make those digestive enzymes and just mooch off of the digestive enzymes made by this bacteria. And so basically, this bacteria might be the stomach for this bacteria. Okay. Those are some examples. There's lots of reasons two bacteria might need each other, but those are just a couple of examples of why you might need a helper species to cultivate a particular bacteria in a lab. Another way we've been able to cultivate once unculturable bacteria is by recreating their entire ecosystem and figuring out ingenious ways of sterilizing the ecosystem of all bacteria but the one of interest. And the reason we do that is, I gave you that list of environmental variables that we consider when trying to culture bacteria in a petri dish, and there were 5 environmental variables on that list. But there could be environmental variables that are important that we don't understand, that we don't know anything about. And so sometimes we just throw up our hands and we'll say, well, we'll just recreate the whole environment, and I don't know what's important here, but at least we'll capture it if we just recreate the whole thing. Right. And many human pathogens include bacteria that typically reside in soils or other environments besides the human body. So this is important for bacteria that we pick up from our environment. But also there are a very large number of pathogens that cannot be cultured in a dish because they require human tissue to live. So for example, Treponema pallidum, which is syphilis, that's an example. But there could be countless other agents of infection yet to be identified due to the fact that we have yet to culture them because they cannot live outside the human body. So there's a number of pathogens that we know cannot be cultured outside the human body, and there could be many others that are mysteries to us, even though they're common infections, just because we can't culture them outside of human tissue. So in this way, concepts in microbial ecology have really helped microbiologists and pathologists get a handle on some unculturable pathogens. And this is one of the reasons I think it's really important to understand microbes as a whole and not to just focus on currently understood pathogens, even if you're going into the health sciences. For one thing, microbiomes are important, but for another thing, by understanding bacteria as a whole, it can inform you on the pathogens that you're studying.